Stanford University.
So it's my great pleasure to introduce Dr. Jake Lusis. I've known Jake for a long period of time, since I was a intern resident cardiology fellow and PhD graduate student at UCLA. Jake has had I would say, a tremendous impact on at the UCLA School of Medicine and also within the human genetics department, as well as in cardiology. We like to claim him, or back then we like to claim him as part of part of UCLA cardiology because of his involvement. So by way of background, Jake received his bachelor's degree in Washington State University. He told me today that he grew up in Tacoma. He then did his PhD in Oregon State University, followed by postdoc in Roswell Park Memorial Institute. Jake is really, in my opinion, one of the similar figures in terms of developing this field called systems genetics. It's not systems biology, it's systems genetics, in which he's trying to integrate epigenomics, transcriptomics, genomics, proteomics, metabolomics, to understand how does the environment and the gene play together. And within the setting of cardiovascular disease, it's very, very well known for establishing this mouse hybrid diversity panel, which I think you'll hear part of that today. He's also taken his discoveries and research to the clinical realm. And I think they have this collaboration, which I'm actually quite interested in hearing. It's 10,000 Finnish men known as the metabolic syndrome in men, or METSIM. I'm not sure if you're going to talk about that, but it sounds very interesting. And then I think the important part of it is that the mouse diversity panel has also translated into our understanding of atherosclerosis, diabetes, insulin resistance, heart failure, and coronary artery disease. Dr. Lusis has published 500 plus papers, most of them in top science journals. I won't read over all of them, but I was very humbled by this CV right here: Cell, Nature, Cell Metabolism, Nature Genetics, JCI, PNAS, and so forth. And it's just list after list of high impact journals. He is also on the NIH board of scientific counselors for the NHLBI, and also on the International Scientific Advisory Board for the Wellcome Trust Center for Human Genetics. He's very well funded. He's currently the PI of a couple all-1 grants, as well as a big program project grant. And then I think many of his trainees have gone on to become faculty members, both at UCLA and elsewhere. And so I could go on, but I think, the guest is the focus. And thank you, Jake, for coming. And we look forward to listening to your talk.
Oh. Well, thanks for that very kind introduction. And thank you for coming today. It's been a long time since I've been at Stanford, and I want to say that this Cardiovascular Institute is just totally amazing. I've had a wonderful morning and so on, and it's also great to see some former students and old friends here at Stanford. Is there like a pointer, or should I just point with this, or? If there's a pointer, oh, you? Yeah. Oh. Thank you, David. Yeah, yeah. Perfect. Okay. Okay, so as Joe mentioned, we're interested in sort of systems genetics, and I'll explain that a little bit. Actually, it's kind of embarrassing to talk about it here. I've been talking to people this morning, and it's sort of like this old saying, bring coals to Newcastle, because so many people here at Stanford are indeed doing systems genetics. And let me begin by just talking a little bit about common cardiometabolic diseases, and sort of as an introduction, I guess. Is this or I guess this guy here. Okay, So I'm just going to read this a little bit. This is one view of common cardiovascular and metabolic diseases. All natural populations will exhibit thousands of common functional genetic variations, and we know that now, that's a fact. And thus, biologic processes will have many different solutions, if you think about it, right? So in general, at least for 99% of us, these will all be good enough, and but under certain conditions, let's say stress, such as an obesity or decreased efficiency, aging, certain solutions will tend to fail. So, and my good friend Jim Weiss, who's chair of cardiology at UCLA, likes to call this the good enough solution. I think Jim spoke here a month or two ago. So now, if all of us in this room here differ, in susceptibility to heart failure or atherosclerosis for 1,000 different genetic reasons, as well as environmental factors, it's clearly a very, very complicated problem, how you're going to address this. And so, to my mind, there are two basic approaches. One is to study in great detail, using biochemistry and molecular biology, the processes that we think are likely to be involved. Let's call this the biochemical approach. And the other is to study the differences among individuals, right? To look at populations. And this would be, let's say, call it the genetic approach. And so, for better or worse, we've been mostly on the genetic side, right? I mean, a lot of people here, I think, are sort of somewhere in the middle. You can use genetics, and then you can also apply molecular tools and so on. So we're doing genetics, and no, I'm sorry. Let's see. Does this move forward with this guy? No. Yeah. Okay. So one approach, obviously, in genetics that you can take is to look at very large populations of individuals and to carry out genome-wide association studies. So in this case, you just look at clinical phenotypes, you look at DNA variations, and you ask if it's very simple, you ask if there's an association. And in this way, you can identify loci. These are generally, these are DNA variations across the genome, and each dot represents a genetic variation. And you correlate it with the clinical trait, you associate it with the clinical trait, and then you plot this as a negative log base 10 of the p-value. And if you exceed a certain threshold, it's likely that that locus contains a gene that contributes to the complex trait, right? So we participated in GWASs, but we don't do GWASs so much. We do another kind of population genetics, which we call systems genetics then, again. And in systems genetics, we also look at populations, but generally much smaller populations. And I'll be talking about human populations and mouse populations. And we look at the clinical traits or traits relevant to the clinical disease, but we also look at molecular traits, particularly traits that we can examine using high throughput technologies, such as expression arrays, RNA-seq, proteomics, metabolomics, that sort of thing, right? And so, so what we do then is we try to relate these molecular traits, not just the DNA variation, but these molecular traits to the clinical trait. And basically we use correlation, we use mapping. Did they map to the same place? And we use modeling, various kinds of modeling. And the idea is to not only understand the locus that contributes to the trait, but how that DNA variation can contribute to sort of molecular traits, actually the flow of information, if you think about it, right? How does information flow from this DNA variation all the way to the clinical trait? So that's the idea. And so what I'm going to do today, it's kind of a general talk that I'm going to give. Oh, I'm sorry. This is, okay, I was going to sort of tell you about some ongoing projects in the lab that illustrate the systems genetics approach. Okay, and first I was going to talk a little bit about following up genome-wide association studies, and I think you've probably all heard about this. It's being done all over the place. It's by far the most common application of systems genetics. And I was just going to briefly tell you about some studies that we're doing along that line. And then I'm going to tell you kind of an unusual application of systems genetics, and that's to identify novel endocrine factors. How do tissues talk to each other? Right? Using basically the molecular information across genetic diversity. And then finally, I'll talk a little bit about host-gut microbial interactions. I know at Stanford here, you have a lot of people that are very much involved in studying the gut microbiome and how it contributes to disease. And I'll tell you how we're trying to apply systems genetics to this problem. Okay, so this first project that I'm going to tell you about is primarily the work of Mete Cibilek. Mete was a postdoctoral fellow in the lab, and about a year ago he moved to the University of Virginia, where he's an assistant professor. Oh shoot, why does it do that? Okay. So following up GWAS, so there are a whole lot of loci for cardiovascular and metabolic traits, right? You can classify them here. There are loci for cardiovascular disease, blood pressure, myocardial infarction, not so much for heart failure, that sort of thing, about 250 loci. Loci for type 2 diabetes, of course, cardiovascular risk is a major cause of disease and death in diabetics, and these measures such as obesity, and so anthropomorphic measures, right? So when you think about GWAS, it's important to remember that it's only a locus, right? It's only a locus. Usually it contains multiple genes. So in most cases, we don't know the causal SNP. There's a peak SNP, right? But we don't know that that's really the cause. It's just the one that we see. Maybe there are SNPs that are variations that we don't even know about. We don't know the gene generally, right? In this locus, there are obviously a lot of genes, right? Here's the peak. It goes across, this is this region of linkage disequilibrium. And we don't know what tissue that causal gene acts in, right? Generally, or what cell type. And you may have a guess, but you don't know. And we don't know for the vast majority of these GWAS loci how they contribute to disease, right? So this is where systems genetics can be applied. And the population that I'm going to tell you about where we're trying to follow up on GWAS is what Joe mentioned. It's this METSIM population. It's called metabolic syndrome in man. And it's a population from, here's a picture of Kuopio. It's a rather small city in eastern Finland. And this is a cohort has been collected entirely by our collaborator, Markku Laakso. He's seen every single individual in that cohort. And so he started collecting this about 10 years ago, and he collected about 10,000 men aged 45 to 70 at the time from this area. And people ask, well, why men, right? Well, in genetics, you try to simplify things. In retrospect, it probably wasn't a good idea, and when we applied for a grant and stuff, we always get this people, well, why not women, right? But I mean, there are other studies in women. We're trying to collaborate. Well, there's a study in the UK called Mother. That's all women, and so we're trying to integrate the two. But it's a random sample of men, right? Not selected for anything in particular. At the 5 to 6 year point, there was a follow-up on this. 7,000 of these individuals came back, and we have very detailed information on those individuals. So it's a longitudinal cross-sectional study, right? And as I was mentioning in talking about systems genetics, it's characterized in great detail. I guess, I don't know if you guys can see this. here, I guess, rather light, but yeah. So there are a lot of clinical traits that have been examined by Marcu, altogether about 250. A lot of these are correlated, but you know, there are detailed interviews, measures like body mass index and so on. Marcu has been particularly interested in type 2 diabetes, so a lot of measures of that, including a glucose tolerance test, but lipids, inflammatory markers, atherosclerosis, heart failure, kidney function, non-alcoholic fatty liver disease, and so on. So it's a whole set of traits that you can try to relate. And then, as I was mentioning, the idea of systems genetics is to not only examine clinical traits, but also molecular traits. So it's been genotyped, and we imputed it to the 1,000 genomes reference genome, so there are like 9 million SNPs that you can compare. It's actually part of it's about half of it will be sequenced. We have adipose tissue from about 1,400 individuals. We've done expression arrays and RNA-seq on about 800 of those individuals. MicroRNA on a subset of individuals we did some time ago. We're doing methylation. We're using reduced representational bisulfite sequencing. A lot of metabolomics on a subset of those individuals, and we're doing the microbiome. So far, we've examined 900 samples using 16S sequencing, and the goal is to get about 3,000. So again, classic systems genetics. You look at all these molecular traits, look at clinical traits, and try to relate them. So what I'm going to talk to you about, just very briefly, is just how one integrates transcript levels, global transcript levels with GWAS loci, right? If anybody's interested later or after the talk or something, I can tell you about methylation and the other traits. Okay, so just as you map clinical traits, obviously you can map molecular traits, right, to particular loci. And so for here, example this is data from about 770 individuals in adipose tissue from this population, right? And so we've done expression arrays. We've now done RNA-seq, but the data I'm showing you is expression arrays. So you can see for this gene here, XRRA1, that it's clearly affected by the genotype at this locus. This is a polymorphism variation, SNP variation, very near this gene. And you can see that there's a clear impact on the expression, right? It's not the whole of the expression. There are other factors that affect expression levels, but clearly has a major impact. And this is called an expression quantitative trait locus, or EQTL, right? And these are very common, actually. If you wanted to look in enough individuals, probably every single gene in the genome would be affected by genetics. Here's a gene, an example of a gene that isn't particularly affected by genetic variation. So there are two kinds of EQTLs that I'm going to mention. One is local or cis. You know, the term cis means that the variation affects the gene on the contiguous DNA strand. That's what cis means. But it's generally local. We assume that they're cis, right? So this could be a variation in an enhancer of a gene or a promoter of a gene or something else that affects the expression of that gene. And then there's transregulation. So in transregulation, you have a locus that affects the expression of genes at other distal sites, right? So for example, if a transcription factor was regulated by this locus here, presumably in cis, it could affect the expression of genes at other loci. So that's the sort of thing that we look at. Okay. Okay. So I mentioned that there are about 680 loci. And so what we did was we asked if peak SNPs or near peak SNPs had EQTLs, and in about 110 of those loci, there were EQTLs at the peak SNPs that regulated gene expression, right? So this is a way of prioritizing the genes at that locus. If you have several genes at a locus, you don't know which one it is. This identifies a functional variation. And then you can further examine whether that is likely to be causal for the disease. What we do, initially we did is this conditional analysis. If you condition on the transcript levels, do you destroy the relationship between the SNP and the disease, right? Just very simple conditioning. You can do other kinds of things. There's this algorithm, summary, data-based Mendelian randomization. You can do, there are a number of sort of causality programs. In my opinion, they don't really work all that well, but you can sort of get an idea. Here is a gene, a large functional variation, and it sort of fits with that being causal for the disease. So it's just simply a way of prioritizing. It doesn't mean, we have these 49 genes now where we have what we feel is reasonably good evidence that a particular gene is causal for the disease. You know, probably we're making some errors here as well, but it now allows us to go further, right? To carry it, to validate it in mouse models and look at it in other ways. And so this is just expression. We can also look at these genes using epigenetics. We can look at metabolites. If there's a gene, is it likely to contribute to variations in a particular metabolite, and so on. So this is systems genetics. And I'm not going to really say much more about CISI, these local EQTL. And I think many people here at Stanford and the Cardiovascular Institute are doing very similar work. We also identified trans-EQTLs. We identified, I think, altogether about 6 or 7 trans-EQTLs that we're very confident of. And we can identify trans-EQTLs because we have a lot of power. We have 770 adipose samples. And I'm just going to tell you about one, which I think is the most interesting one. And it's a trans EQTL locus that was actually identified by Karen Small and Mark McCarthy several years ago. And it's at a transcription factor called KLF14, right? And in this publication, they showed, or they presented evidence that variation of the KLF14 locus affected the expression of about 10 different genes, 10 loci. And they also found that those genes that were regulated in trans tended to have relationships to metabolic diseases, right? And so now with our data, we're collaborating with Karen and Mark, and we're characterizing the KLF14 locus. And now we have more data, so we can say more about what it does in trans. So now if you add the additional data, there are about 55 target genes shown in this circa plot here. Here's KLF14. Here are the genes that it regulates. With this p-value of 5 times 10 to the minus 8. And if you actually do a Bonferroni for a trans regulation, it's 10 to the minus 13. But a lot of them reach 10 to the minus 13. So there's no question that this is a trans locus. And so there are about 55. If you want to do an FDR of 5%, there are almost 400 genes. So this is a locus that clearly impacts a whole lot of different genes, right? It does a lot of things. Now, so we've done we've looked at mice. I'm not going to really talk about that, knockout mice and so on. One of the things that we've looked at is the nature of the variation of the KLF14 locus. So here, there is a cis regulatory variation at that locus, and presumably that's what's causing the trans regulation. So there are here's a polymorphism that is associated with altered levels of KLF14. And there's a haplotype that goes with it, and it's in what's called a stretch enhancer. And it's just a name. It's like Nick and I were talking about this. It's just a sort of a complicated enhancer. It has many different elements. And there's a certain haplotype of this enhancer. And so what we've done is examine it in an adipocyte tissue culture cell to see if it fits with what we find in the population. And so the haplotype associated with high expression does indeed have higher enhancer activity in vitro. It's kind of an interesting variation. It's absolutely adipocyte specific. KLF14 is expressed in almost every tissue, but this polymorphism only affects expression in adipocytes, nowhere else. It's also restricted to Europeans. Right? This polymorphism doesn't occur in Asian or African populations. What's really interesting about KLF14 is that if you look at the 55 genes that are regulated by KLF14 and you ask, is their expression correlated with metabolic traits, there are a lot of, this goes from -0.4 to 0.4, so this is very, very significant. Something like 0.3, 0.2 is very significant association. So it's associated, these are the genes, and these are some of the clinical traits from the METSIM that KLF14 is associated with. So it really goes, so this is truly sort of a metabolic syndrome master gene, right? Okay, so let's see. I was going to switch now. So that's just sort of an illustration of sort of the systems genetics approach. I don't know, are there any comments or if you have any questions, please feel free to interrupt. Any questions or comments about this approach? No? Okay. But feel free to interrupt. Okay, so the second example I'm going to show you of application of systems genetics is one that is entirely the brainchild of Marcus Selden, who's a postdoc in the lab. He just finished his second year as a postdoc in the lab. And we have this mouse resource that I'll tell you about. And he came in and he was thinking about how it could be applied. And he came up with an idea that it could be used to identify novel endocrine factors, particularly proteins that are involved in communication between different tissues, right? So we know a lot of endocrine factors, right? I mean, these have mostly been identified by, sort of looking in plasma for proteins or certain proteins, diabetes, and isolating pancreatic cells and so on. So mostly biochemical methods. And so this is a different approach. This uses just systems genetics data. The way that it sort of works, I'll show you. Oh, I'm sorry. Let me go back. How do I go back here? Is there a way of going back? Can I go back with this? Oh, I can. Okay. So here is the idea. Okay. So in populations, things vary. Almost all genes vary in the population. Could be for genetic reasons, environmental reasons, but everything varies, right? So let's say that now this is either a human population or a mouse population. I'm going to talk about the mouse because this works much better in the mouse. So here you have a putative endocrine factor. It's secreted and it acts on a separate tissue, right? Now, in this population, we'll have variation of gene X if we can monitor it in a particular tissue, let's say by expression arrays or proteomics, okay? And if indeed it's acting on a separate tissue, that you might predict that there would be a correlation between the expression of this gene and a set of genes in the target tissue, right? Might predict. It doesn't have to be up and up and up, it can be up and down or whatever, right? But you might expect that there would be this correlation. And so I really wouldn't think that this would work very well, but Marcus tried it and it works very well. So Marcus has now identified, well, he screened maybe a dozen candidate genes and identified like 5 novel endocrine factors. And I'm going to tell you about one of these, okay? So the population that we look at is very well suited for this because it's a population of mice that are studied under identical environmental conditions. And this is this population that Joe mentioned. It's called the Hybrid Mouse Diversity Panel. And it's a big name. So it's just about 100 commercially available inbred strains of mice, right? You can buy them from Jackson Labs. There's nothing special about them. You can raise them under different environmental conditions, but apart from that, nothing special. They are highly diverse, so there's a lot of genetic variation. About the same amount of genetic variation, common genetic variation, is in the human population. we originally constructed this to do genome-wide association studies in mice and to study gene-environment interactions. But now it's characterized for, since we set this up in 2010, for atherosclerosis, obesity, diabetes, heart failure, fatty liver, bone density, dietary responses. So we have a whole bunch of clinical traits on these across the 100 strains. We've also looked at molecular traits. Again, this systems genetics idea. So we've looked at transcriptomic, epigenetic, proteomic, metabolomic data in selected tissues. You know, in some sort of surveys under certain conditions, we've looked at more or less, but we have data on multiple tissues. And so Marcus thought, well, if we have data on multiple tissues, we have transcript levels in, let's say, adipose and some other tissue, let's see if this works. And so he set this up. And so the method is simply this, is for each secreted protein in the sender tissue, let's say the tissue that you think is producing the endocrine factor, you ask whether the correlation with genes in the target tissue is greater than expected by chance. It's very simple. And what he does is he just takes the negative log base 10 of the p-value of the correlation, and it grows from the candidate gene across all genes in the target tissue. Okay. So let's say we have this situation. We have data in the hypothalamus. We have data in fat tissue, and we're predicting now, we're going to look for endocrine factors that are produced by fat that act on the hypothalamus, right? So what we do then is for each gene in fat, we calculate an average correlation of genes in the hypothalamus. And if we do that, we get kind of a normal distribution. This is the average correlation here, average negative log base 10 of the p-value. It's kind of a normal distribution and somewhat skewed in this direction. If we now filter for secreted proteins, and there are about 2,000 secreted proteins in adipose, this is shown here, right? We get this. Now, can anybody think of a gene that might be involved in communication between adipose and hypothalamus? Okay, right. Okay, better know this, right? So leptin, of course. We know that leptin is produced by adipose tissue. It's secreted, it signals to the hypothalamus, it affects food intake and energy expenditure. And if we look at leptin expression in the HMDP strains, we see that there's variation, considerable variation, like threefold variation, and not, surprisingly, it's associated with adiposity, right? Fatter mice have this. So if we look now at leptin, where does leptin fall on this curve? Is it significantly more correlated with genes in the hypothalamus than, well, than most genes, right? Than you'd expect by chance. And so what you find is leptin's right here, right? So if you look at, we've done proteomics in the hypothalamus, and if you look at the proteome level, leptin is the #1 gene. It's the very top, most correlated. If you look at expression data, it's the #3 gene. And so it's high. So now we've done this, Marcus has done this across a whole bunch of tissues. So far we've looked at a data set that's where the mice have been fed a high-fat, high-sucrose diet, but we have other data sets. And so in that cross, we have looked at, we have expression data or proteomic data in adipose, hypothalamus, liver, intestine, and muscle, right? So pairwise, you can just look at all of these tissues. You can identify the top-ranking genes and see what they tell you. So this is the top 5 in going from adipose to hypothalamus, adipose to liver, and so on. And what you'll find is that a lot of these genes here that are in green are already known endocrine factors, right? So this is reassuring, right? Because we're here finding things that you expect. Here's leptin. This is by expression data. It's #3. But a lot of these are not known, right? The ones in white. I also actually have some of these bolded here, and there's some evidence for the genetic variation affects clinical traits in those. So what we're doing then is looking at the ones that haven't been discovered, right? That's what Marcus has been doing. So he's picking some. He's been particularly interested in interaction with muscle and in insulin resistance. So he's picked some of these genes, and like I mentioned, I think he's looked at about 12, and he's validated. I'll show you how he identifies the gene and what it does, but he's validated, I think, like 4 or 5 now. So I'll show you how that works. Okay. Oh, so I should say, okay, so the one that I'm going to tell you about is this LCN5. It's lipocalin 5. And lipocalin, it's never been shown to be involved in communication between adipose and muscle at all. There are about 4 papers on it. It's a member of a large family. It's secreted into the circulation, but really what it does is some of the other lipocalins have been studied in more detail. This one, nothing. Okay, so now one of the tricks that Marcus uses is that he picks genes that are expressed primarily in one tissue. Like if lipocalin 5 was expressed in half a dozen tissues, you wouldn't know, what's sending and what's receiving and stuff. So he picks genes that are expressed in one particular tissue. This is a log plot. Here you can see. So the expression in white adipose is much, much higher than any other tissue. Okay. So now, what might it be doing? It's affecting. It's correlated with the genes in muscle, skeletal muscle. But why? Oh, I'm sorry. Let me go back here. So what he does is he takes the genes that are being perturbed, most by lipocalin. In this case, he picked 500 genes, and he just puts it into David. And I don't know if you know what David is. David contains a whole bunch of pathways and processes and so on. And you can just ask, is this set of 500 genes enriched for certain pathways or processes, right? And when he does that, he finds mitochondria, 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 right? So it looks like it's affecting some metabolic function. So one easy thing that we can do in the HMDP, because we have all this clinical data, is lipocalin 5, is it associated with anything that would be related to mitochondrial function? And so this is just a lookup, right? Just a lookup. And so what we can see in our HMDP, this is 105 inbred strains, that it is indeed significantly associated with body weight, visceral fat, sub-Q, gonadal fat, and fat mass, right? BICOR is just sort of like a variation of Pearson correlation where you sort of normalize the data and you take out outliers. So it's a very significant relation. So all this fits. So now he moves on. And again, you can do this at very high throughput. He just expresses the protein with a tag. He expresses it in a cell line, pulls it down, he's got an isolated protein, and now he adds it to tissue culture cells, and he asks, does he see an effect? And this is a skeletal muscle cell line called C2C12. And so what he did was pick a bunch of proteins, transcripts, that are involved in mitochondrial biogenesis and oxidative gene expression. This is just some of these. And here, if he adds recombinant LCN5, you can see it activates these. Okay. This is one experiment. There's a lot of variation, but it's very, very reproducible. There's no doubt it works. You can also do in vivo function, or sort of functional tests on these cell lines. So for example, there's something called the Seahorse bioanalyzer, which really just measures oxygen consumption. So you plate the cells in the Seahorse, and you ask if LCN5 affects respiration. So you can look at basal respiration, or if you uncouple, then you can look at maximal respiration. And indeed, LCN5 does affect respiration. Again, consistent with the function in mitochondria. And then we've done some in vivo studies. We haven't knocked out LCN5. That would be something to do. But we can express it with adeno-associated virus, and a particular virus that's constructed in a way so that it's expressed primarily in adipose tissue. So we put LCN5 into this adeno-associated virus and infect mice. And here I'm showing you one thing. We've done glucose tolerance tests, insulin sensitivity, and so on. But by all these criteria, it works. So it does indeed affect insulin sensitivity significantly, right? This is the change in glucose following that injection of the insulin. So it does function in vivo. And then, of course, one thing you would want to know is it doing anything in humans, right? So we obtained human myotubes, primary myotubes. And here again, the human homologue of LCN5 in mouse is LCN6, it's called in human. And so we add purified LCN6 now to human myotubes, and you can see that there are these same kinds of genes. Mitochondrial biogenesis, oxidative phosphorylation genes are indeed induced. And then we can go back to our METSIM. So in our METSIM, I mentioned we have adipose expression for about 770 people. And so we can simply ask, is LCN5 expression in the METSIM correlated with the traits that we would predict, such as insulin resistance? And you can see that there's a lot of scatter, but again, we see a significant correlation between the transcript levels of LCN5 and the Matsuda index is an index for insulin resistance, right? So as I mentioned, Marcus has now examined a number of these. So I think, you know, it's how far do you go or something like that. But it clearly is a good way to identify novel endocrine proteins, endocrine factors. And what we'd like to do, of course, we're interested, like you all here in the Cardiovascular Institute, we're interested in heart. And so we have other studies where we've looked at heart function. We've looked at an isoproterenol model for heart failure, and we've looked at atherosclerosis. So these are, we have lists of genes that we're going to look at to try to identify factors that affect heart disease. So I think this is the last slide of this one. Are there any questions or comments on this? Yeah, Joe.
So you show a slide where it's an organ.
Huh?
How do you sort it out that it's only tons of other
Well, you know, probably there are, you know, I think we know lots of endocrine factors, but I think it's just the tip of the iceberg. I think there are hundreds of factors that remain to be identified. So very likely that tissue is communicating in multiple ways, but we've identified one of the ways. And then what we go back is by expressing that protein and administering it alone, we see a similar phenotype, right? That's the idea. But it's a very simple concept. It's just correlation. Yeah. Any other comments? No? Okay. So the last part of this is host-gut microbiome interactions. And this is the work primarily of Eileen Org. Eileen was a postdoc. And she's now back in Tartu, her native Estonia, where she's an assistant professor. And Diana, she has been a long-term researcher in the lab. So this is primarily their work. Okay. Now, I think all of you have probably heard ad nauseum about how the gut microbiome is important in clinical traits. And, you know, if you think about it, I mean, we've co-evolved for millions and millions of years, right? Not just necessarily as humans, but we have co-evolved. So it's not so surprising that there should be lots of lines of communication, right? And if there's communication, then there are going to be impacts. And it's not so surprising to me that the gut microbiome could influence clinical traits. Obviously, things like inflammatory bowel disease, but even other traits like autism and so on. And so about 6 years ago, as part of our sort of systems genetics thing, what we thought, well, it's pretty easy to monitor the gut microbiome. It's very simple with sequencing. And so why don't we monitor the gut microbiome? So we started looking at it in our mice. We started looking at it in human populations. So this just shows, these are phyla, actually. And this is phyla this is in 113 mouse strains, part of our HMDP. And this is in 200 of the METSIM population here, right? And you can see in both cases, firmicutes in blue, Bacteroidetes in red are the major phyla that are present. There are other phyla, and you can see in the lighter color here. There's a lot of variation. Now in this case, the mice were on exactly the same diet. They were bred for at least 2 generations at UCLA, so why do they differ so much? And so because we have the genotypes, the genomes of all the mice, we could calculate heritability. And it's kind of surprising. The heritability, if you do this in mice, for most phyla and genera, is between 0.3 and 0.5. So the host has a major, major impact on the kind of bacteria that are in our intestines. But diet is also, diet actually, I would say, trumps genetics. So here it is in humans, and that obviously varies for all kinds of reasons, dietary reasons and genetic reasons and so on. Now, if indeed gut microbiota are important in clinical traits, common clinical traits, then there should be a relationship between the composition of the gut microbiota and the clinical traits. And so we've used that to try to identify some bacteria, species, genera that contribute to clinical traits. And I'm just going to show you one real quickly here. So this is Brian Parks identified, part of his study was this microbiome study, and there was a strong correlation between Akkermansia mucinophila, which is a bacteria that grows on the mucin layer of the gut, and insulin resistance. And so what we did in that case, just to check it out, then in a couple cases now, is we grew up, we cultured Akkermansia, we gavaged it into the mouse by just sticking a tube down, and we gavaged it with either heat-killed or alive Akkermansia. And you can see that it has an impact on cholesterol, has an impact on body fat, but most strikingly, it has a very large impact on insulin resistance. Right? And this has been confirmed, very similar results. We use this strain AXB19, but very similar results from another laboratory. So I think it's real. And so this is just an example. This is sort of, what at the time was sort of to convince us that really there are these effects. And we've done other kinds of things. We've, for example, we've done cross-fostering, switching the bacteria from one strain to another strain. And again, you can see these kind of effects. So the bacteria in our guts are doing things. They're doing things that affect metabolism and so on. But the big question is how they're doing it. And so although there have been many, many publications on gut microbiota and so on, really we know very little about that. We know, well, there's some factors. We know short-chain fatty acids are produced by bacteria. They can affect blood pressure and so on. Bacteria act on bile acids. Bacteria help the immune system to mature. But our understanding of all this is very, very minimal. So I think a major discovery came with the discovery by Stan. And I think Stan, I don't know if many of you remember, Stan told me he was here about a year ago, I think, and talked. So Stan made this really very important finding that he did an unbiased screen. Again, this is systems genetics, right? This is what systems genetics is. So he screened about a small number of cases and controls for heart disease, and he found that trimethylamine N oxide was associated. Trimethylamine N oxide, as I show you, is derived from the diet through the action of gut bacteria. He went on and confirmed this in a larger population. And then he knew we'd been studying mice and looking at atherosclerosis and so on. So he contacted us to see if we had a cross. We sent him the plasma, and very strong correlation between trimethylamine N oxide and atherosclerosis in mice as well, right? So the way that it works is that choline, and subsequently he found carnitine as well, are converted by gut bacteria to trimethylamine. This is actually a gas that gives fish its very sort of fishy smell. This is absorbed in the circulation. It's acted on in the liver primarily by flavine monooxygenases, primarily flavine monooxygenase 3, to produce trimethylamine N oxide, which then somehow contributes to heart disease. Right? So we've been collaborating with Stan since then. We've been doing the microbiota part, and we've been looking at this FMO3 in some more detail, and so that's what I'm going to tell you about. So since that original study, there have been a lot of other studies that, I wouldn't say they also are the same thing, but they're at least consistent with the role of trimethylamine N oxide in heart disease. Okay, oh, this is from one study. So this is where you divide individuals by TMAO level into quartiles. And you can see this is a prospective study. And you can see that the individuals in this highest quartile have a much greater likelihood of suffering a myocardial infarction or a stroke. Okay. So is it causal? That's been one question. Stan in his original studies just administered mice TMAO, and it did increase atherosclerosis, so it seemed likely. We've been looking with this enzyme FMO3 that converts TMA to TMAO. And so what we did was we used an antisense oligo that it's very effective in vivo. And this is the protein level. This is a Western blot. And you can see we can really knock down FMO3 very nicely. And when we knock it down, as expected, we see an increase in TMA, trimethylamine, and a decrease in TMAO. And we also see a decrease in atherosclerosis, which is consistent. This isn't a perfect study because the knocking down FMO3 also has some effect on lipids. Okay, so we're trying to follow up. But it's certainly consistent with a causal relationship. It isn't TMA. TMA is also associated in populations, but it looks like it's TMAO. Okay. So how is it acting? So now we go back to the METSIM population. So we've looked at about 1,000 people here for TMAO levels. And, you know, I showed you all those traits that it's correlated with, or that we've looked at in the METSIM. And it doesn't go with anything except one, which is glomerular filtration rate. It does go with that. So there may be some impact of TMAO also on kidney function. Or maybe it's the glomerular filtration rate that affects TMAO levels, right? So we don't know that. So what does it do? So we were studying mice. We were feeding them a high choline diet, and we decided to look at the aortas, take out the aorta from the mice, and see if we could see differences in gene expression as a result of the choline diet, right? High choline diet. And what we found, here's what the effect of the choline diet. Choline goes up a little bit, TMA goes up, and TMAO goes up quite a lot. I thought it was going to be early, I'm sorry. So what you can see is a bunch of inflammatory markers also go up. Markers, genes, chemokines, cytokines, adhesion molecules associated with atherosclerosis. And so what we found is, we treated the mice with TMAO. We found those same things going up. All of these are in fact regulated by NF-kappa B. So we looked at NF-kappa B signaling. These are just Western blots. And this is the quantitation. So we think that what's happening, one of the things that TMAO is doing, these are not super high concentrations. Some individuals have concentrations this high. What we think is doing is TMAO is activating the NF-kappa B pathway in smooth muscle cells and endothelial cells. It works in both. In cultured endothelial cells or cultured smooth muscle cells, it does the same thing. This activates the expression of inflammatory genes, adhesion molecules, and that contributes to atherosclerosis. We don't know the receptor. We're actually trying to find the receptor using systems genetics because some strains of mice respond more to TMAO than other strains, right? So conceivably, you could map it. Dan just had this very nice paper recently where he showed that TMAO also potentiates platelet activation and promotes clotting. So this panel, what TMA doesn't activate platelets by itself. If you give thrombin, there's a release of calcium into the cytoplasm, and you can detect it with this fluor, right? If you give thrombin, though, and there's TMAO present, it greatly potentiates the action of thrombin, right? Very dramatic. And actually, you can see the TMAO has a function, a clotting function in vivo. There's an iron chloride assay for coagulation. It induces coagulation. And if you give mice TMAO or you feed them choline, coagulation time is decreased. So that's another factor. It affects coagulation. And in fact, I won't show you data, but it also affects adiposity, right? On a high-fat diet, we have knockout mice now for FMO3. There's a clear impact on, I don't know, I think it's TMAO. It may affect kidney disease. There's some evidence that it affects heart failure. So it's like a whole bunch of things are affected by this microbiota-derived metabolite. And in my opinion, this is, Stan may have talked about this before, but you know, there are hundreds of metabolites that are produced by microbiota, and there are these what are called uremic toxins, like indolyl indoxyl sulfate, or there's several of these guys, right? And so they're very interesting candidates. So I think that this is just the tip of the iceberg. There are likely to be lots and lots of metabolites, really dramatic interactions between diet and disease. So I think that's about it. Well, you know, one of the nice things about this, exciting things about this, is that microbiota present an opportunity for you know, sort of intervention, right? One can use small molecule inhibitors, and Stan actually has developed some inhibitors. There are about a dozen different bacteria that produce TMA, at least a dozen, probably more. But they all use the same enzyme, so Stan has developed an inhibitor that inhibits all of them, right? But you can do other kinds of things that you know, you've heard about this stuff before, probiotics and so on. What we're focusing on is very complex. So we're trying to understand the relationship between genetics, diet, and disease, right? So it's this tripartite thing. It's very, very complex interactions. Diet has a huge impact. I showed you genetics. Diet has a huge impact. So in addition to being a substrate for production of trimethylamine, choline dramatically impacts the composition of the gut microbiota. This is principal component analysis, sort of a summary of the composition. You can see huge effect, and all kinds of dietary components do this. And of course, you can identify the bacteria.
Stanford University.